Okay. Okay, good afternoon, everyone. I'm pleased to see so many people have logged on. I hope it's helpful for you to actually have this live class as well as the recorded class. Today we're going to start a series of classes on microbial metabolism. Many of the principles that you're going to learn in this section on metabolism will apply to eukaryote metabolism as well. There are a couple of special things that in prokaryotes that we'll hear about, but most of the metabolic processes that we're going to talk about are shared by eukaryotes for the simple reason that most of these processes actually evolved in prokaryotes and have been inherited by the eukaryotes as evolution proceeded. This just suggests to you that many of these processes, in fact, probably evolved in the last universal common ancestor to all three of the living groups, which are the prokaryotes, this should say eubacteria, by the way, the archaea and the eukaryotes. Remember that viruses are not living, they have no metabolism. So they are not included in our discussion. This is simply there to stun you. And this is actually a tiny little version of a huge chart, which you can obtain if you want, which covers many square feet of the wall, detailing all of the metabolic processes in a living cell. And it's simply there to impress on you how very, very complex these metabolic processes are. That's the first thing. But second of all, how they are all interlinked. Because we have to employ some sort of reductionism, we talk about particular metabolic processes, but we need always to remember that the metabolic processes that we talk about actually feed into all sorts of other processes as well. The metabolic functioning of a living cell is unbelievably complex, but we can boil it down to it being about dealing with matter and dealing with energy. This is the cell's way of dealing with matter and energy, of growing, for example, by building molecules, by getting energy, by breaking them down, etc., etc., etc. So what we're going to have to talk about are these aspects. In particular, what does energy mean to the cell, and how does the cell use energy to manipulate matter in the cell? And so the first things to get straight are we're going to be talking about two broad categories of reactions inside the cell. The first are called the anabolic reactions. These are the reactions of synthesis. You've probably heard the term anabolic steroid, for example. An anabolic steroid is a hormone which enables cells to build mass, for example, to build muscle. So an anabolic reaction is one of building, of creating, for example, for polymerizing a molecule. That is an anabolic reaction. And it's a reaction of synthesis. It takes simpler things and it puts them together to make something more complex. In order to do that, the cell has to have a source of energy. It is an energy demanding process. We say, therefore, it is endergonic. It requires an input of energy. And it is usually done by a dehydration synthesis. We usually make the larger molecules, we join them together by abstracting a water molecule each time to form covalent bonds and to make the larger molecule. We'll see many examples of this. The other set of reactions are the catabolic reactions. And these are the reactions of breakdown, of breaking large molecules, for example, large polymers into smaller units, breaking covalent bonds, and very often doing so not only to get rid of a molecule, the cell may need to do that, but these are the reactions which the cell performs in order to extract energy. For example, the cell may have made large storage molecules like fats, lipids, etc. And now want to access that, access the energy in those large molecules. In order to do so, the cell is going to have to break bonds. It's going to have to break down those molecules. And when it does so, it does it in a careful, stepwise fashion in order to extract the energy. And very often, as we'll see in a second, to feed that energy into these anabolic reactions. So these catabolic reactions are reactions of breaking and they usually are done by a process of putting in a water molecule. Where here in anabolic, we took out a water molecule in a dehydration reaction, bonded them together. Now in catabolic reactions, very often the water molecule is put back in and the bonds are severed and energy is extracted. So that is called hydrolysis, the process of breaking by putting in a water molecule. So these, of course, do not sit on their own. I've already hinted, these reactions feed into one another in almost a cyclical fashion. The energy and matter is moved around the cell. Complex molecules in the anabolic reactions require an input of energy. Where is that energy taken from? It's taken largely from fuels, which are broken in catabolic reactions. The energy is put into the molecules to build them. Other large molecules are broken down in catabolism, in catabolism, in order to fund the anabolic reactions. So these diagrams here give you the summary of the two. Macromolecules are broken down into simpler molecules in catabolic reactions or catabolism, and energy is released. And very often that energy is going to be harvested, and it's going to be put into a form which can be fed to the anabolic reactions. And the anabolic reactions are the opposite. They take simple molecules, bind them together by covalent bonds, and those covalent bonds use energy to form them and to form the larger biomolecules. So that immediately gives you this idea that these processes are ongoing in the cell the whole time, and we're already talking about matter. We're talking about building and breaking molecules. We're talking about changing the nature of matter in the cell, and we're talking about shepherding in energy back and forth. We're taking energy and we're storing it, then we're extracting it, putting it into these reactions. What we're talking about is continual transformation of matter and energy within the cell. So what we're going to find, all of these reactions are often very complex. The very first thing to say is that very few biological reactions will occur spontaneously. Will occur spontaneously at any fast rate. Most reactions, biological reactions, complex reactions, are extremely, under normal circumstances, if we did them in a test tube, they would be extremely slow, too slow to be useful to the cell. The cell has got to have ways of speeding these things up, of making them much more likely to occur. Now, the cell does most of these complex reactions, not in just one fell swoop, but in series of steps. So have a look here and we see, okay, we've got a large molecule, right? here it is here. Maybe this is starch or glycogen or something that the cell wants to break down and extract energy from, something like that, perhaps. It doesn't just shatter this and end up being able to extract the energy. Instead, it pushes this initial reactant through a whole series of reactions. Here's one reaction here, here's another reaction here, here's another reaction here, here's the final reaction here. At each of these points, an intermediate product is produced. Here's one here, here's one here, here's one here, here's the final product. This is very useful for the cell and for all sorts of things. First of all, it provides the cell with ways of controlling the reactions. Second of all, these intermediates may themselves, although there is a final product at the end, these intermediates may be useful. They may be useful in other reactions. And that is one of the ways in which all of that complexity arises in the cell of different metabolic reactions feeding to one another. This is one of the ways they do it. That's the first thing. And the second thing, sorry. First thing, control. Second of all, maybe feeding into other metabolic pathways. A single set of reactions like this may be useful in several different ways. So this stepwise is called a metabolic pathway. And it has another characteristic, and that is that each of these reactions takes place by the intervention of its own specific enzyme. And an enzyme is a substance, a biological substance, which acts as a catalyst. It speeds up the rate of reaction. Remember, I told you, most of these reactions in biological systems under normal circumstances occur so slowly that they would be of no use to the cell. The cell needs a product now, on demand. When it's needed, it needs to be produced very quickly. The way in which the cell does that is by having enzymes which facilitate each of these steps. And the whole reaction, the whole series of reactions in the metabolic pathway may occur very rapidly because of the intervention of enzymes. This also goes to the point of that the cell can exert control over this whole pathway by controlling the activity of its enzymes, either by controlling when the enzymes are produced. Enzymes are, most of them, 99% of our enzymes are proteins, and they are produced from the genes, from the recipes locked into the DNA. So the cell may control pathways by controlling the production of enzymes to govern the pathways. There are various other ways we'll hear about later on as well. Okay, so if we're talking about, especially about energy, there's some very special considerations to think about inside the cell. Energy doesn't just fly around like lightning bolts from one side of the cell to the other. The energy has to be very carefully controlled, very carefully dealt with. Energy needs to be stored, for example, so that the cell has a constant source of energy. When nutrition and everything is abundant and extra energy can be produced, that energy can be put into a storage compound somewhere. But then when there are demands for energy, the cell needs to be able to access that energy, and it needs to be able to put it into the places where it is needed. That is not necessarily an easy thing to do. And in fact, has some complexity to it, because it has to be a carefully directed process. It's no good having a store of energy if you can't access that energy and put it right where it's needed. Okay, you're even accessing it. is useless to the cell unless it can be directed where it's needed. So these are some of the considerations that we need to think about. There are numerous energy-rich compounds in a cell. And the first set that we need to think about are those which can actually act as fuels. So these are things we've already heard about, things like the fats, things like glycogen. All of these things, what did all of those things, all of those molecules have in common? Well, what they all had in common is they have long chains of a carbon skeleton of one sort or another. Depends on what organism you're talking about. But attached to those carbons, they have many, many hydrogens. And locked into those hydrogen bonds, sorry, not hydrogen bonds, locked into the bonds between carbons and hydrogens is a huge amount of energy. That is the principal source of energy for the cell. The energy, in other words, is derived from nutrients built into storage products like lipids or starch or something like that. So fine, there's our energy store. Those are long-term storage compounds, very stable, and the bonds between carbons and hydrogen, they don't break that easily. We're going to see it actually requires a complex set of metabolic reactions to extract the energy. But once you've extracted the energy, then how on earth do you get that energy to the place where it's needed? The cell can't just crack a bond between hydrogen and carbon and release energy. And what's it going to do? No, it has to be put into a form which the cell can use. It has to be put into a biologically useful form of energy. And the principal biologically useful form of energy is energy locked into this compound here, ATP, which is adenosine triphosphate. And its name tells you everything you need to know, because it is an adenosine molecule, which we'll see in a minute. There it is there. And it has attached to it 3 phosphate groups here. 1, 2, 3. Adenosine 1, 2, triphosphate. Adenosine triphosphate is made by the cell from adenosine diphosphate with just 2 phosphate groups, adenosine plus phosphate plus a phosphate. And the cell adds a phosphate group to it. And there are different ways in which this can occur. There are different ways that the cell can actually perform this action. Adding a phosphate group is called phosphorylation. And we're going to see 2 principal ways of forming ATP, or by phosphorylation. We're going to talk about first a set of chemical reactions where we just add it in. And that is called substrate phosphorylation. But there is another much more important cellular process called oxidative phosphorylation. And that occurs in the process of cellular respiration, where we use oxygen actually in the process. That's why it's oxidative. So we're going to hear about 2 principal ways at the moment, and there's substrate and oxidative phosphorylation, whereby the cell adds in a phosphate to ADP and forms ATP. So here's the rub. There's the energy. Where does this energy come from? Well, in heterotrophic processes, it comes from a source of fuel, a fuel source of some sort. Okay, and this substance, ATP, is the principal way in which the cell takes energy and makes it available to a metabolic pathway, to a specific set of reactions, is in the form of ATP. That's the most important biologically useful form of energy. There are one or two others you will sometimes see, but in general, ATP is 99% of the energy transfer in a cell, uses ATP as the ultimate, as the donor to the reactions. And all of that energy that is used lies here in this bond here. It's the last of the bonds. It's actually rather unstable. So ATP spontaneously, quite easily breaks down and releases this energy. So you may, in one or two reactions, see this bond used as well to ultimately produce adenosine with just one phosphate on it, but that's not very common. In most reactions in the cell, most metabolic reactions, we see adenosine triphosphate dephosphorylated to adenosine diphosphate. And the energy of this bond is what is used for whatever purpose the cell needs energy. So ATP is the most important energy currency, But we've got to think about this. And that is, what is the actual, we just say, oh, energy. We put in energy here to make this bond. We actually have to think about, where does that energy come from? I've already suggested it to you. It is energy which has been locked into those hydrogen bonds. It's actually locked in the hydrogen atoms that are attached most prominently to carbon atoms in the forms of fuels and things like that. So we've got to think about how do we get the energy, this powerful chemical energy in a fuel, in that carbon hydrogen bond. How do we take that energy and how do we put it in here? It's not going to fly across like a little lightning bolt and just go in. There has to be a process to transfer that energy, to convert the energy in actual fact. And it is done by these intermediate substances, which we call cofactors. And these are substances which act as electron carriers. So most of the energy is locked into the electron of that hydrogen, and it's extremely energetic. It contains a huge amount of energy. When we break that carbon-hydrogen bond in a fuel, what we're going to do is we're going to break it, and we're going to transfer electrons with all of their energy to these substances. The proton comes with it, by the way, temporarily, but we'll hear more about that later. So the first important one is nicotinamide adenine dinucleotide, NADH. And in its ionic form is NAD, or actually NAD+. And when we want to extract energy from a fuel, what we're going to do is we're going to make NADH. There's the hydrogen joined onto it. It's acting as a carrier. What is really important is that it's carrying the electron, this hydrogen electron here. We're going to see how that is used. This is the other one, important one, is flavine adenine dinucleotide. And this what usually occurs in the form FADH, and when it's carrying a hydrogen, FADH2. Again, here it's carrying a hydrogen, carrying a proton, but it's, most importantly, it's carrying that hydrogen electron with all of its energy. And these carriers actually contribute that energy of the hydrogen to this reaction. So what we've got these two systems. We've got a system making ATP, and we've got a system for extracting these energy-rich electrons and transporting them to this reaction. We're going to spend a lot of time talking about how the cell actually does this, because these are the processes of breaking fuels and shepherding that energy to useful forms of energy, of ATP. Okay, so this is just, this basically is just a summary. I just want to point this out to you. Here is this really, really significant molecule, which is ATP. And have a look here. Here's a double ring structure. And here's a sugar, it's a ribose sugar. And here are phosphates. Wait a minute. Where did we hear about this? Where did we hear about sugar and then phosphates and these ring structures? We heard about it when we talk about nucleic acids as macromolecules, right? This is actually a nucleotide, and it's almost identical to a nucleotide, which gets included in the nucleic acids. And because this is, we call this, when it's free like this, adenosine, and it is adenosine triphosphate. But look here, there's the adenine, which we see in the nucleic acids. It's exactly the same. It's just being used for a different purpose. So this is actually a nucleotide, but here it's being used as an energy transporting mechanism. It's not a very big molecule, but it carries with it a huge amount of energy here. And many of the cell's processes are dedicated to making ATP, to fund all of the energy requiring reactions of the cell, everything, building molecules in anabolism, movement. All of these things require a constant input of energy, and it is provided in the form of ATP. So yeah, these are just basically summary slides for you. Energy is released from ATP when we break that hydrogen bond, and it is actually used in metabolic reactions. The way in which the energy is transferred from ATP to the reaction is by transfer of, very often, of this entire phosphate group here, together with the energy of the bond. So that is the way in which the energy will be transferred. We dephosphorylate. I'm sorry, I've got the little things in the way here for me. I don't know about you. But for example, if we need to put in energy to a reaction, we need to break the ATP. Very often that bond is broken by transferring the entire phosphate group to the reactions, often through them, usually through the intermediate activity of the enzymes. And we effectively dephosphorylate, produce ADP by breaking that bond and using the energy in reactions. Here's an important point though. This molecule ATP is very unstable. It is usually very short-lived. It's continuously, this is why it's continuously produced by the cell, because almost as soon as it's produced, it needs to be used. That this bond is easily broken, and the energy is immediately extracted. So this means this is short-term. This is the most useful form of energy in biological systems for funding reactions. But of course, the cell needs to have long-term storage as well. And these are the fuel molecules that we've talked about before. In animals, in eukaryotes rather, it is usually in the form of these huge molecules like glycogen in the eukaryotes, or starch. In prokaryotes, it's usually in the form, the storage forms are usually glycogen and phospholipids. And lipids, not phospholipids, but lipids. And glycogen by far the most common, a long-term storage. These are stable molecules. But because they are stable, they require a considerable input, in a considerable amount of metabolic activity to actually extract the energy, to break those bonds. We're going to talk extensively about how we do that, especially in the processes of respiration. So in eukaryotes, by the way, they have a little bit more versatility for storing energy, because plants, for example, use starch. Animals use lipids and glycogen, both. But in the prokaryotes, there are two principal storage products. There's another storage product, I'm sorry, apart from the lipids and glycogen, and it's this substance here called poly-beta hydroxybutyrate, and often just referred to as PHB. The only reason I mention that, we're not going to talk a lot about polyhydroxybutyrate, but a polyhydroxybutyrate is often accumulated in the cytoplasm of prokaryotes and actually appears as crystals. And you'll remember we talked about inclusions before. Those inclusions these polyhydroxybutyrate crystals often appear as inclusions in the cell. Glycogen, by far the most important of the long-term storage products for prokaryotes. Okay, so this is just a comparison of the two of these processes of anabolism and catabolism. And remember how strongly they are linked to one another. Anabolism, synthesis. Catabolism, breakdown. Anabolism, the ultimate products, usually large molecules of one sort or another. Catabolism breaks large molecules into smaller, simpler components. Very often takes a polymer and breaks it into its monomers. A typical anabolic reaction, which we haven't mentioned much, is photosynthesis. And that is photosynthesis is the third way of producing ATP. Photosynthesis is also referred to very often as photophosphorylation. It is a way for plants, or for photosynthetic organisms rather, to manufacture ATP, and it is taking the energy of sunlight and putting it into ATP. It then uses the ATP to make molecules like glucose. So that is photophosphorylation. So remember, now we've got substrate phosphorylation, oxidative, which we will talk about later. That's the processes of cellular respiration, and photophosphorylation, the processes of photosynthesis. In the breakdown of fuels, extracting the energy from fuels, here are the major processes that we're going to be talking about. We're going to be talking about things like glycolysis, breaking of glucose, and all sorts of other cycles. Never mind about this, it won't mean much to you at the moment, but there are all sorts of metabolic cycles of catabolism for extracting energy from fuel. All of these are mediated by enzymes. All of these processes. Virtually none of the processes occur under controlled fashion unless we have enzymes present. Anabolism requires an input of energy. They are endergonic. And catabolic reactions will release energy from the reactions. They are exergonic. Okay, so let's just think for a minute. We're not going to talk extensively about the actual chemistry of these kinds of reactions, but I just want to remind you of some basic chemical principles, because it begins to impinge on what we are going to talk about. The first is to think about why a chemical reaction happens at all. What we've been talking about so far, the transfer of the energy, changing the matter, these are all chemical reactions. But why does a chemical reaction occur at all? And it occurs because atoms in one form or another actually interact with one another. And they shift. They actually very often are shifting electrons from one place to another. And in doing so, they are often forming bonds or breaking bonds or whatever. So in order for atoms to interact with one another, they have to be physically proximate. That's the first thing. The second thing is that when they contact one another, they need to contact with one another with enough energy that the reaction can actually take place at all. And that the energy required for them to break bonds, for example, and reestablish bonds, and et cetera, this is an amount of energy which we call the activation energy. It's in the amount of energy required for a reaction to actually occur. Now, under normal circumstances, all atoms and compounds, they are moving with a certain amount of kinetic energy. And that the amount of kinetic energy is a function of the temperature. If two atoms smack into one another with enough energy, they can rearrange the distribution of the electrons, et cetera, and reorganize their structures. So, for example, here's a simple one. This molecule here is methane. It's a carbon with 4 hydrogens attached to it. And methane is actually a household gas. It's a fuel. It's actually I will just mention on the side that it's actually largely the product of bacterial activity, the production of methane. But we use it as a household gas. Okay, so here it is. We're going to take it. In order to do anything useful with household gas, well, you've got to light it. You have to put it with oxygen and give it enough energy, and it will burn, and it will produce 2 products. It will produce carbon dioxide, and it will produce water. Have a look here, and you'll realize that we have exactly the same atoms present. Here are the reactants here. Here are the products here, carbon dioxide and water. We've got exactly the same atoms there. What has happened is that they've just been rearranged. And they've been rearranged into new compounds. In order to do this, these individual atoms have to smack into one another with enough energy. They have to collide with one another with enough energy. So here, this is not methane, but yeah, I've got a lighter. I hope it's still got gas in it. And I'm releasing a little bit. Okay, so I've released some now into this room. Now I'm going to ask you this question. I've released gas, fuel, into the air, which is full of oxygen. Are those molecules that I released, are they burning? Yes or no? Well, they're certainly not producing a huge flame. They're not producing the useful byproducts of heat and light, which is what we expect from the burning of methane. But the answer is yes. In actual fact, these molecules will drift off, and every now and again, occasionally, they will hit into an oxygen molecule with enough energy that they will effectively burn, and they'll produce carbon dioxide and water. But they do not do it sufficiently fast or in sufficient quantity for it to produce a noticeable effect. And the reason for that is that it occurs very slowly. When I just release the gas molecules and I let them go off, every now and again, one will get hit by a cosmic ray and acquire enough kinetic energy to react with oxygen. Okay, so on and so on and so on. But in order for anything to actually happen that's useful, what do I have to do? Did you see? I had to put in some energy. I put in a burst of activation energy. And I get the whole clump of these to burn all at once. And once they burn, then they release enough energy that it activates the oncoming molecules. The reaction now becomes self-sustaining. There's my spark, the gas coming out. There's a little spark. And once I set it going, then it becomes self-sustaining. It requires that input of activation energy to get sufficient of them to react that it produces a useful reaction. So here's an important principle in biological systems. Most of the reactions that we're going to talk about, they will occur spontaneously, okay? But they occur so slowly, so few of them have sufficient energy to actually react, that it is biologically useless. If we want a set of biological reactions to take place, we have to encourage the collisions. We have to encourage their proximity, and we have to force molecules to interact under circumstances which are favorable for the reaction to occur. So what we need to do in biological reactions, we need to increase the reaction rate. And what is limiting the reaction rate is this activation energy. Yeah, because they require a lot of activation energy in order to react. So the way in which we do that in biological systems is by utilizing, it could happen if we increase temperature or pressure or whatever, but that's very difficult to do in a biological system. In a chemical system, you can do it. You can increase the reaction rate by increasing the temperature. Why? Because you increase the kinetic energy of the atoms and they smack into one another harder. Biological systems, it's very difficult to do that. Most biological reactions take place within very narrow temperature limits. Same with pressure and everything else. But in biological systems, the important way in which we increase the rate of reaction is by utilizing enzymes, by utilizing these biological substances which will catalyze a reaction. And we will spend a little bit of time talking about enzymes and about how they work. So an enzyme is a biological. Yes, go ahead.
Sorry to interrupt, but I have a question on next week. Do we have a quiz?
Yeah, please save these kind of questions for the end, because it kind of interrupts the train of thought. No, we don't. You have a test next week. You don't have a quiz this week. You have a test next week. All right. Okay.
Thanks.
Sure. Okay, so here's a biological system. Here it is here. We've got two reactants. These biological systems, the reactants very seldom will react spontaneously with one another at a rate which is biologically useful. Instead, the reaction rate is very, very slow. And the reason for that is that the molecules are simply not in a situation where they have a tremendous amount of collision energy. In order to get these molecules to react with one another, we've got to put in energy, which is the activation energy. You have to put in energy to get them to react at all. And for most biological molecules, that energy of activation is actually quite high. So that is the barrier. It's because they require this activation energy, this high activation energy, that the overall reaction rate is very slow. And it's so slow that it's biologically useless. Instead, biological systems make use of enzymes, and the function of an enzyme is to lower the energy of activation. When an enzyme is present, it takes the reactants and it puts them into a situation, usually a physical situation, where the reaction is made much more likely. For example, the enzyme may take two reactants, separate reactants, and it may force them together. And at the same time, it may utilize energy which has been provided to it by phosphorylation. And it takes that energy, forces the reactants together, and makes them react with one another. And then it releases them. And then the enzyme redoes the same thing with the next set of molecules. It does it over and over and over again, very rapidly. Enzymes function to speed up the reaction rate. They do not make impossible reactions happen. They work on reactions which are possible, but which would normally happen very slowly. But they lower the energy of activation. So here in this graph here, here's the activation energy there. Here's the activation energy without enzyme. Here it is with enzyme. It's lowered. And because it's lowered, the reaction rate increases. And it increases to biologically useful proportions.
I have a question.
Yes, go ahead.
So what creates the activation energy?
It's inherent to the nature of the reactants.
Okay.
Yeah, that is a function of the structure and the chemistry of the reactants. Also of the environment, but in biological systems, the environment in general is kept fairly constant. It's not like doing a reaction in a test tube. But in biological systems, most of the biological reactions occur very slowly under normal circumstances without the enzymes.
Thank you.
Sure. Okay, so here we're going to talk a little bit about how these enzymes actually do operate. I hinted to it with my grasping at things in the air, that the enzyme is a physical object. Okay? It has a physical structure. Most of our enzymes are proteins, and they are extremely complex in their structure. But they are folded in a three-dimensional shape with one particular area of the enzyme, which is strongly reactive to very specific substances. Enzymes have great specificity, and they will fit only to particular substances. If it's for example, an enzyme which is promoting, let's say, an anabolic reaction, the enzyme may fit two substrates, and it only fits those two substrates, but it fits with great specificity, and they have great attractive ability for those substrates. That is a function of these particular areas here of the enzyme, which is called the active site. That is the place that is the business site of the enzyme. This is where it binds to the things which it needs to react to, in one way or another. Here in this example here, they are using an enzyme called sucrase, and sucrase does is it actually binds sucrose, and sucrose is two sugar molecules joined together, and it's glucose and fructose joined together. They are different molecules. When they separate, they have different structure. So this active site has a site to bind one side of the sucrose molecule, and this side is the other side of the sucrose molecule. And so here it is binding sucrose, the substrate. Once this happens, that the binding creates a compound. It's a compound between the enzyme and the substrate. We refer to it as the enzyme substrate complex. Once that happens, this induces change in an actual shape of the enzyme itself. And the enzyme actually moves to stress this bond here. between the glucose and the fructose. It actually causes this bond, physically causes it to break. And once it's broken, the enzyme loses its ability to bind the substance. The enzyme binds the entire sucrose molecule. It doesn't bind the individuals, so individual components. Once those components are formed, it releases them, glucose and a fructose. And the enzyme now returns to its original shape, and it can do the same thing over again. Enzymes can operate for a very long time, and they perform these reactions extremely rapidly. So they take substrates, they force things to happen to those substrates, and then they release the product. And then they return to their original conformation, and they are able to do the same thing over again. Okay, that's just the same thing, sorry. You can use that for your revision. Okay, so if we were to look at this in chemical terms, here's what we'd see, okay? So this we'd present as this somewhat dull drawing of this chemical structures of sucrose. This here is fructose, this is glucose. And this is another name for, I used the word sucrase. It's also called invertase, it's the same thing. Here it breaks the two into the two components. But don't think about it in these boring chemical diagram forms. Think about it in the active biological sense. This enzyme here is a physical entity, and it is grabbing substrate and it is forcing it to do things, and then it's releasing products. So in this here, what is the substrate? The substrate is sucrose. What are the products? The products are glucose and fructose. What is the enzyme? The enzyme is the invertase. And remember that with somewhere along the line, we might actually be able to make sucrose. Plants certainly do, and they do it by taking fructose and glucose, using an enzyme to put them together. Okay, so the possibilities, both possibilities are available to the cell. Depends on the pathway, depends on the enzymes, and it depends on the purpose. Okay, let's just take a break for 5 minutes, and then we will carry on.
I just had a quick question on the difference between like the cofactor and the coenzyme.
Sure.
I just don't really understand like the concept of like what it means.
Okay, is there, first of all, a coenzyme is usually organic. It's a organic substance. A cofactor does not have to be organic. And a cofactor is usually a non-protein as well. What would I say? A coenzyme, let's just say that the coenzyme is something that actually temporarily forms part of the functional enzyme. Okay, I don't think I'm explaining this very well, and I think I need to think about it for a minute. You know what I'm going to do? I'm going to go to your textbook, and I'm going to read up exactly what they say about it so that I don't mislead you. And please remind me at our next, if I forget, I will send out a little note clarifying this, all right, because I have not explained it well. Okay, so leaving that aside for the moment, I will send out a clarification. Usually when we're just talking about enzymes, you can immediately recognize that a substance is an enzyme because the last 3 letters are ASE. It implies that this is an enzyme. So the oxidoreductases, which we're going to see many of, are enzymes which facilitate oxidation reduction reactions. Now remind yourselves about what an oxidation reaction is and what a reduction reaction is. A reduction reaction actually adds electrons, and it usually adds electrons to a substance. And those electrons are usually added in the form of a hydrogen atom. So you're taking what they're actually interested in is the transfer of the hydrogen electron. But in biological systems, we usually see the transfer of the whole hydrogen atom. That may be temporarily separated, as we'll see in a while. You may actually separate off just the hydrogen proton, which is H+, which you remember, if I add H+ to a solution, I'm going to lower the pH, I'm going to increase the acidity, okay? But a reduction reaction adds electrons. An oxidation reaction takes away electrons. So the two reactions go in hand because we've got to have a source of electrons. We get the electrons through oxidation reactions. We reduce substances by adding electrons, which we've derived from oxidation. It's important to get this, to remember this, because we're going to, as we start to talk about processes of respiration, especially, these oxidation reduction reactions are really important. Transferases actually take a functional group. You remember your functional groups like the hydroxyl group and carboxyl group, et cetera, et cetera. A transferase will actually take a whole functional group and move it from one substance to another. There are some specific names for some of the transferases. For instance, there are phosphorylases, which are also transferases, which take phosphate groups and move them. And those would be important in phosphorylating enzymes, for example. Hydrolyses, there are many different hydrolyses, but they catalyze reactions of hydrolysis. In other words, they're extensively involved in catabolism, because hydrolysis is the principal way in which we break up polymers. Isomerases actually rearrange isomers. So they will take a molecule and they will twist it into a new formation, for example, to create a new isomer. Ligases we're going to hear about extensively when we talk about DNA and DNA function. Ligases actually join molecules together. And this is an energy requiring reaction. And they, of course, it's an anabolic reaction, and so it uses ATP. All right, so let's just talk a little bit about influences on enzyme activity. We accept that the enzymes are actually lowering the energy of activation and speeding up the rates of biological reactions. But enzyme activity, remember, most of the enzymes are proteins, and they are very much affected by environmental conditions. So, for example, temperature, pH have a marked effect on the functioning of the protein enzymes. But there are a couple of other things which we will also discuss. And one of them to think about is, what is the influence of having different concentrations of substrate? If we push a whole lot of substrate into a system with enzymes, does it actually affect the rate of the reaction? So that we'll talk about a little bit. And then the last thing to think about is, there are ways in which enzymes can actually be controlled. There's one principal way in which the cell controls it. And this is by use of processes of inhibition, actually deliberately slowing down enzyme activity. So let's have a look first of all at the influence of temperature. Very high temperature, remember most enzymes are protein, and protein is very much affected by temperature. And at very high temperature, protein may be permanently denatured. It loses its activity to act at all. So very high temperatures, this is why very high temperatures are damaging to most living systems, because it denatures their proteins. But below that denaturation level, what is the influence of temperature? Well, it's exactly the same kind of influence, really, that we'd expect in any chemical system. If as we increase the temperature, we actually start to increase the rate of collisions that are naturally occurring. But at the same time, that facilitates the activity of the enzymes. And up to a certain point, increases in temperature actually increase enzyme activity. So we have a look here, and you'll see in this temperature range, 10 degrees to about 37 degrees Celsius, there's a steady increase in the rate of reaction in the enzyme activity to this point here. And that is called the optimum point. And this is the temperature at which this particular enzyme has its maximum activity. That is a function very often of the structure of the enzyme and the nature of its activity. But for many biological systems, around about 37 degrees Celsius is the optimal. So for us, for example, we maintain a constant body temperature of around that. And almost all of our enzymes, their optimum is here at 37. Below the optimal, their ability to work is less. Above the optimal, the enzyme actually begins to get damaged and very rapidly loses its ability to operate at all. So the rate of reaction is strongly influenced by temperature, but most living things, first of all, if they are able to, like we are, they will maintain a constant temperature at which most of their enzymes operate. Other organisms which are dependent on external temperature will often regulate the temperature by actually moving to areas where the temperature is optimal for them, for all of their enzymes. I will just mention that prokaryotes are very, very diverse in the temperatures at which their enzymes will operate. This is not typical of all prokaryotes, for example. There are prokaryotes whose enzymes operate optimally at very low temperatures. And there are a very few prokaryotes whose enzymes operate at very high temperatures, above boiling point of water in actual fact. They have specialized enzymes, but their enzymes operate at a very high temperature. It depends on the organism. It depends on what environment they have evolved to occupy. Their enzymes are suited to that particular environment. Okay, so pH also has a tremendous effect on enzyme activity. And to understand why, there are several reasons why, but one of the reasons is that most of our enzymes are proteins. And just think back to the structure of proteins. How are proteins made? They are made from long polypeptide strands. And all of their folding, a lot of their structure actually depends on ionic and on hydrogen bonding, which stabilize a particular shape in space. And that shape is what determines the enzyme activity. In addition, in the active site of the enzyme, a lot of the reactions that take place there are because of charged side groups to the amino acids that are there, the formation of temporary hydrogen bonds and all sorts of, all of that bonding ability is strongly influenced by the amount of protons that are present in the solution, the amount of H plus that is present in the solution. The amount of H plus is what determines the pH. So we find that enzymes have an optimum pH for most biological systems, not for all, but for most biological systems, the optimum is actually just below 7. In this little graph that they've given you here, the optimum for this particular one is around about 5. But just remember that in actual fact, most biological systems, the optimum is around about here. Okay. So there is an optimum pH at which the enzyme functions best. Below that, it begins to lose ability. That can be for various reasons. It can be because as you change the pH, the shape of the enzyme actually begins to change. Or it can be because as you change the pH, the function of the active site is affected. Its ability to form bonds becomes affected. So the optimum, again, if we exceed the optimum, we run the risk of actually denaturing the enzyme. So almost all living things very carefully regulate their internal pH. But things can go wrong. And for example, there are disease situations in human beings, for example, such as acidosis, where the body pH begins to fall very low. And one of the reasons that is so dangerous, such a dangerous situation, is because of this, because the enzymes that are driving metabolic reactions begin to be inhibited by the low pH. Or sometimes it can be that the body solutions actually become more alkaline. Same thing happens. The enzyme activity is actually impacted. There is an optimum. Each enzyme has an optimum pH at which it operates. And living systems do everything they can to maintain those optimum conditions. Okay, so here's the other thing which might affect enzyme activity. And that is to increase the concentration of the substrates molecules which the enzyme reacts together. Well, it will seem logical that as we increase the substrate concentration, the enzyme can work faster and faster, better and better, but only up to a point. There is a point, so what we'll see is there's a very rapid increase in enzyme activity as we increase the substrate concentration. But there is a point at which increasing the substrate concentration no longer makes any difference. And that is because the enzyme activity becomes saturated. There's a point at which filling the active site, producing product, filling the active site, producing product is happening at a maximal rate. And adding more substrate is not going to increase the enzyme's ability to perform the reaction. So we see this typical graph here with a very, very steep exponential increase in activity, and then a leveling off once the enzyme becomes saturated. Okay, so now we need to think about this. Enzymes operate by having active sites which are very, very carefully constructed. They are very carefully constructed to fit to a substrate, and then once that fit has taken place, to perform a certain action, very often stretching bonds or forcing bonds to occur or adding a phosphate or whatever. The enzyme, its first step is to fit the substrates and to fit them exactly into the active site. There are various ways in which we can impact the ability of the enzyme to actually do that binding. The first way I'm going to tell you about is a completely artificial way. This is what I mean by that is this is not a way in which the cell ever uses to control enzyme activity, but it's a very important one to think about, especially in disease processes. And this is called competitive binding. So here's the normal binding of the substrate to an enzyme. Okay, maybe this was our sucrase, and this is sucrose, okay? And it's binding here, and it binds very, very specifically to the molecular structures of the substrate. There is a system called competitive inhibition. And in competitive inhibition, a substance comes in and it fits in to the active site. All it has to do is fit into part of the active site, and it completely inhibits enzyme activity. Why? Because we now have this substance sitting here and the substrate cannot bind, because the enzyme binds the whole substrate. It doesn't just bind part of it and act. It has to bind the whole of the substrate. But here part of the active site is being occupied by a competitive inhibitor. These competitive inhibitors very often bind irreversibly to the enzyme. Once they're stuck there, they do not leave. And this is a typical situation for toxins and for poisons. So this is not a natural system that the cell would use to control its own enzyme activity. But this is something that can happen to an organism if it becomes poisoned. So for example, a simple example is the chemical cyanide. Cyanide actually binds to the active site of a respiratory enzyme, and it completely stops cellular respiration from occurring because that enzyme cannot operate. Okay, so that's one. The other one in microbiology becomes very important are that there are many clinically significant microorganisms which release toxins. For example, botulinum. Botulinum toxin is released by an organism called Clostridium. There are various Clostridia, but Clostridium botulinum produces this toxin, which is paralytic. It actually binds to enzymes involved with muscle contraction, and it paralyzes the muscles. Botox, for example, people have injected into their foreheads or wherever. Botox is a paralytic toxin which binds to enzymes and stops muscle and paralyzes muscles. So it's really important to realize these are substances which come in from outside. They are not produced by an organism in order to control its own enzymes. That is competitive inhibition, and it is usually irreversible. Okay. I will just briefly tell you about non-competitive, and then we'll stop, and I'll return to this slide next time. The alternative inhibitory system is called non-competitive inhibition. And this is a way in which cells can control their own enzyme activity. It works in a little bit differently. Here's an enzyme, and here's its active site where it's going to bind its substrates. But many enzymes have another site. It's not an active site, but it's a site remote from the active site, but somewhere on the enzyme. And it's here. It's called the allosteric site. There's the word there, the allosteric site. And it will bind a substance. And when it binds the substance, it actually changes the shape of the enzyme, and the enzyme is no longer able to bind the substrates. So, well, how does the cell use this why have this system at all? Well, think about it this way. How about if I have these, this enzyme is performing a reaction in the cell in order to supply the cell with glucose, let's say. This is sucrase. It's breaking sucrose to provide the cell with glucose, okay? So there it goes. How do we stop it when there's enough glucose? Well, one way you could do that would be by having this allosteric site bind glucose. If we got enough glucose in the cell and we bind all the allosteric sites with glucose, then the enzyme will stop working. It'll stop producing glucose. Okay, the cell will use up the glucose, and as glucose levels drop, the allosteric site will become free, because the allosteric site, this binding is by no means irreversible. It's very reversible. It gets reversed very easily. And so this is a way very often we'll see in metabolic pathways, the very first enzyme in the pathway is an allosteric enzyme. And what controls it is the final product of the metabolic pathway. As the cell produces enough of the final product, it inhibits the metabolic pathway by stopping the very first enzyme. So this is a very important cellular mechanism, allosteric inhibition or non-competitive inhibition. Okay, so I'll stop there for today. Does anybody have questions? Feel free, please, to.
One question. So what time should we come for the exam Wednesday? Is it at 4 or 5?
Yes. It'll be set for the class period. And all you do is go to Canvas and go to quizzes and hit the quizzes button, and the quiz you'll see the quiz there. It'll pop up, and it's all multiple choice.
Okay, and then for when I go on the MyLab after I did the assignment, it says consider reviewing assignment 1. Do we do that one too?
Yes, that will come up if you're below a certain percentage. It's not absolutely necessary, but if it's short, it's usually short.
Okay.
So you can do it. And by the way, this week's assignment, I think I'll only be able to get it up tomorrow. I think it'll be a bit shorter than last week's. All right.
Okay. Thank you.
I have a question.
Go ahead.
Are you grading the assignment? Are you grading the assignment based on the percentage on MyLab, or are you just giving us credit for doing it?
No, it's the grade that you get. Oh. It's the grade that you get, and don't forget, if you get a low grade on it, then there is that follow-up one, which you can do to make up for it.
Professor, I've been having problems with the
Mm-hmm.
access code, and I contacted customer support, and they told me that all the students used up the access code, so I'm not able to join MyLab.
Okay, I thought that I had this problem, and I thought that they had fixed it up. Sorry, I can't see. Who am I talking to?
Hannah Magana.
Oh yes, hi Hannah. Yeah, you sent me an email, didn't you? Let me get hold of them again, because I thought they said that they had sorted this out. Try again later. Later in the week, I'll send her an email now, and she'll probably do something later in the week. So try Friday, Thursday, tomorrow afternoon or Friday. Try again and see if you can get in.
Thank you.
All right.
Professor, I have a question.
Go ahead.
Just to clarify, so the test is going to be multiple choice on Wednesday, and it's going to be on the first 3 PowerPoints.
Yes, up till today, up till the end of today.
Okay, sounds good.
All right. Yeah. All right, everybody, if no more questions, thank you all for coming. It's very gratifying to see so many people do come to these live sessions. And it is, even though it is remote, it's nice to know that there's living ears on the other end of my ramblings. All right, and I'll see you then next week. Okay, bye.
Thank you, Professor.
Bye-bye.
Thank you, Professor.